Hello everyone, this is Dr. Anjali Bhatt, co-scientific chair of this conclave. A very warm welcome to all of you. I have with me Dr. Shilpa Shirore, who would be introducing the next speaker and the topic. Over to you, Dr. Shilpa.
Thank you. Good afternoon, everybody. It is an honor for me to introduce Dr. Shiv Sarin. He is an honorary professor at the Jawaharlal Nehru Center for Advanced Scientific Research. He is also a former chairman for the Board of Governors of the MCI. And it is an honor to tell you that he has been awarded with the Padma Bhushan Award by the Government of India. Over to you, Doctor.
Thank you for inviting me, and it's a great pleasure to join. Can I see the first slide, please? Or is it someone else to speak? Sorry.
No, sir, it's you to speak, but we are waiting for the slides to be shared. We'll take a moment for the slides to get over here. Now we have the slides, sir.
I bring greetings from the Institute of Liver and Biliary Sciences, and that's a very hot topic which you have given me, and I always enjoyed IDEC meetings. Next slide, please. I have nothing to disclose. Next. Except that we have very bright faculty. Next. And bright residents. We still rank number 2 in the country after AIIMS Delhi for second year in a row. Next, please. I'll discuss gut microbiota, which is the whole virus, fungus and others, but I'll just cover the microorganism, the bacteria in health, in metabolic syndrome, and some modulations. Next, please. This is an important slide for you to see that there are 5 types: the Bacteroidetes, the Firmicutes, the Actinobacteriaceae, Proteobacteria, and Microbacteria. Now you please, this is from the upper and this is the lower bowel. It shows you an important thing that these bacteria, they make actually the 2 of these make something like 65 to 90% of the total bacteria. These live into a low pH area, as you can see here, compared to the high pH area where the other bacteria are here. You remember that the transition is based on the oxygen requirement of the bacteria. So you have a phylum, you have a class, you have order, family, genus, and a species like E. coli. Next, please. To simplify, and I'll try and make my talk simple, we have 5 types. Now the Bacteroidetes are in the distal gut. They break down carbohydrates. They produce acetate, succinate, propionate. These are the energy sources. Firmicutes, which are in the upper bowel mainly, and Faecalibacterium is common, they make butyrate, another short chain fatty acids. Actinobacteriaceae is a probiotic. Proteobacteria, which includes your E. coli and others, they are opportunistic pathogens. They remain quiet if others are good. And finally, 1 to 4% is your Verrucomicrobia, which includes a very important species which maintains your mucin, Akkermansia, and this is one which makes your intestinal mucosal health. So remember, the bacteria, 5 of these, and 2 are the main ones, and which make short chain fatty acids. Next, please. What do these short chain fatty acids as a fermentation product do? Butyrate gives mucosal energy. Acetate, if it is there, gives you lipogenesis. Lipids in the liver are stored. Propionates, the glucose deposits, and alcohol, the endogenous, not the exogenous, also goes to liver and adds into the gluconeogenesis. Next, please. Now, these are some difficult slides, but I want you to understand these are the basis of the microbial fermentation for last 12 months. When the microbes ferment, they use through the G-coupled protein receptors, the GPCR41, which gives you the peptide YY, which is your appetite sensor. This decreases the gut transit time. And because of this, the bacteria extract more short chain fatty acids, energy, and this leads to obesity because of hepatic and the adipose tissue lipogenesis. Next, please. On the other hand, if you have the G-coupled protein receptor 43, this is 41, this is 43, everything is reversed. You have the white adipose tissue, the muscle, and the L cells in the intestine. They all have a different role, which leads to a obesity prevention, more insulin sensitivity, more improved glucose tolerance. Next, please. So the microbial fermentation, when it is of these short chain fatty acids, is in the adipose tissue and it decreases PPAR gamma, hepatic PPAR gamma. You have obesity prevention, improved insulin sensitivity, reduced hepatic steatosis. Now you will question, how come the same microbes do different functions? And I showed you what does acetate, what does succinate, propionate, and butyrate do. We'll go through it again, but everything is regulated by microbial fermentation. Next, please. So this is a paper which came in Cell Metabolism, just to tell you that the bowel Klebsiella pneumoniae, if there are three types, less, moderate, and high alcohol producer. The high alcohol producer Klebsiella, if this is given to the animal, then it can produce something like a fatty liver disease. So per se, the production by Klebsiella of alcohol can lead to fatty liver disease. Next, please. Alcohol per se, when somebody drinks alcohol, the gut microbes change, and very importantly, you have a reduction in the Akkermansia and an increase in Klebsiella and E. coli. Of course, somebody has alcoholic liver disease, the whole spectrum changes. But this is in a healthy bowel that you have effect on the bacteria and on the metabolites. Next, please. Now, this Akkermansia, if it is in abundance, it reduces weight, type 2 diabetes, and even hypertension. And this is a Nature Medicine paper. When overweight or obese volunteers who were insulin resistant were given Akkermansia 10 raised to power 10 orally, either live or pasteurized for 3 months. Next, please. There are several interesting slides, but I am showing you only one. Compared to control, where insulin resistance increased 0.07, there was a significant reduction when live or pasteurized Akkermansia was given. Insulin resistance came down. Single bacteria makes a change in your insulin and the signature glucose. Next, please. So what all does it do, Akkermansia, which is just 1 to 4%. In obesity, it is decreased. Type 2 diabetes, it is decreased. I don't have time to tell you. But what is important is metformin. When somebody is given metformin and has type 2 diabetes, this bacteria quantity increases. From here you can see here, and it also increases the goblet cell number and the changes in the gut microbiota. I told you this is the one which has the mucin protection, increased mucin production in the intestinal mucosa. So in a way, metformin is a probiotic. Next, please. Now this is an important slide, and based on something like this, the Nobel Prize for 2017 was given. You have a day and night cycle, the light and dark cycle, and your the nucleus in the brain, suprachiasmatic nucleus SCN, regulates this part of the thing. Exposure to sunlight or sleep is the one which determines all your circadian rhythm, the physiological effects of this. What is important is that even during the feeding or the fasting state, the gut microbe and the intestine, they have a circadian rhythm. When somebody is fed, let's say in the night or in the day, the firmicutes increase. and they will extract 35-40% of the calories. On the other hand, if the person is fasting, bacteroides and the varico microbiota, they increase. Fasting is important. Why? These organisms will break down bile acids and will produce secondary bile acids, which are very important for the integrity of the bowel. While this, if you continuously keep eating, you will keep adding to your weight. So it is extremely important what time do you sleep, what time do you eat. If you continuously eat at night, it is not healthy, and this is the whole basis of weight gain. So in the night, the same food eaten versus the same food eaten in the day, the extraction of the calories depends upon the correlation 3:1, 2:1, 1:1. If the firmicutes is 3 and these are 1, you will keep on extracting calories and keep on adding to the metabolic syndrome. Next, please. With this background, I'll give you some idea about the coexistence of gut microbiota and the host. They regulate each other's circadian rhythm. Gut microbiota regulates when you will sleep, and host decides by his habits what time the gut microbiota should go off to sleep. Sleep means firmicutes are sleeping. The dysbiosis is a disruption of this coexistence. An overgrowth of certain bacteria at a given point of time certainly is harmful. The richer your profile of bacteria, higher the diversity, better you are. Next, please. Now, a dysbiosis, as I said, is change of bad over good. What will it lead to? It will allow a leaky gut. The intestines are inflamed. The microbiota metabolites are changed. They will translocate through the intercellular junctions as well as transcellularly. They will get into the blood, and the bile acid composition, more primary bile acids, worse you are. Secondary bile acids, better you are. So the dysbiosis changes our life. Next, please. So in health, you have a thick, good mucin, you have propionate, butyrate, and you have them propionate and butyrate actually gut immunity. The lymphocytes of the gut are very protective. To the L cells, you have GLP-1, you have other PYY peptides, and this decreases the energy intake, decreases your blood glucose. Beta gamma and the oxidation allows an anaerobic environment to be there, and the goblet cells make the antimicrobial peptides and signals. So this is your health. But once you have eaten a lot of sugar, carbohydrates, or sometimes even fats, you will realize these protective things gone down, the mucin layer has gone down, your mucosa associated invariant T cells called mate cells, T regulatory cells, these are lymphocytes, they all go down, and you start extracting more energy, your cells are inflamed, and your glucose levels go up, and of course the antimicrobial defense goes, and this leads to large number of diseases, especially metabolic diseases. Next, please. In very simple terms, having fatty food or having carbohydrates in very large quantity allows a leaky gut. The lipopolysaccharides will go to the liver through the portal vein. You have high TNF alpha, IKKB beta, and an insulin resistance. And of course, the hepatocyte inflammasomes are actually numbed out. They are not able to sense the inflammation. Next, please. Just to give you a simple idea in a patient who is in a subject who is healthy versus cirrhosis and ascites. In a healthy person, firmicutes make about 51%, but what is important is the opportunistic organism, the proteobacteria, are about 5%. But in dysbiosis, they become half. 53% is your opportunistic. So they take an opportunity in a leaky gut, will go into the portal vein, and cause infections and all kinds of problems. And this is true also of many other diseases, including IBD and others. Next, please. The same mechanisms will allow the gut bacteria to get into the liver. The stellate cells become inactive. You have other cells and inflammation markers. I'll not go into details, but just to tell you that liver cancer is a product of this continued inflammation in NASH, and you can stop it by several methods, including giving antibiotics, probiotics, and of course giving the gut a strong barrier, from a leaky to a strong barrier in the gut. Next, please. Next, please. So if I want to define in a simple term, the gut microbes are the signaling hub for normal living, for your immunity, and deciding how much energy you require or you want to spend. If you spoil it, your life and your metabolism is spoiled. Next. How do you modulate it? In the next half of my presentation, I will try and help you how diet, probiotics or symbiotics, and fecal transplant can make a change in gut microbiome. I am not covering the targeted approaches like restoring microbial metabolites, bioengineered bacteria, and different modulation pathways. Next, please. Now, this is within first 2 years of life. When the baby is born, the mother's milk gives oligosaccharides, and you see these are infantile bifidobacterium, very healthy. When the switchover occurs to the diet, which is the oral diet, after this solid foods, you see that the adult strains of bifidobacterium come, and Clostridium and other bacteria come to give you short chain fatty acids to extract energy from carbohydrates, and also it starts giving you secondary bile acids which maintain the antibacterial milieu in the gut. This is the most antibacterial thing. If you have less secondary bile acids, more sepsis will occur. Next, please. Now, this is a very simple study done in 32 of 16 males with a nearly overweight to slight obese propensity. Energy was balanced, carbohydrates for 2 weeks, and then sweetened beverages, fructose was given, 25% of energy for 8 weeks. Now you will see that these people, everybody who received the fructose for 2 weeks, 8 weeks, 10 weeks, they had very high circulating triglyceride. Just to tell you, by giving carbohydrates over 6 to 8 or 10 weeks, fasting sugar, insulin, triglycerides, lipogenesis, hepatic fat, visceral fat increases. So diet per se will change your body's triglycerides and hepatic triglycerides. Next, please. So there are 3 types of scenarios. You have a fiber, you have a high fat, and you have a high meat, the red meat diet. I'll just cover the first 2. The green is the one which gives you the normal fiber diet, short chain fatty acids, decreases blood pressure. The N3-PUFA and the polyphenols, the colored agents, will give you something like fat in fasting induced adipose factor or angiopoietin-like factor 4. This is a very important substance now for us to have a stimulated PPAR gamma, decrease LPLs, and also it will decrease adiposity and the white adipose tissue. On the other hand, somebody has red meat. It has the TMA, the trimethylamine, and its oxidases, which leads to atherosclerosis. Now here, if you have metformin, which all the diabetologists use, it, as I said, increases akkermansia, decreases bacteroides, and it also inhibits the FXR, decreases bile acid synthesis, and decreases liver fat and glucose. So I think this is a drug-induced prebiotic which all of us should use in more than one situation. Next, please. Mediterranean diet given in overweight people and obese people causes reduction in cholesterol, and of course it is through gut microbiome, and it increases substances like ruminicoccus, which is very good for us, includes lactobacilli and others, and the fecal total bile acids are decreased. Next, please. Now this slide I might have shown earlier also. People drink alcohol. Some of them get liver disease and others never get liver disease. And this is dependent upon the intestinal microbiota, which are resistant to endogenous as well as exogenous alcohol. And they keep your liver healthy, no fat, no injury in them. But how do you convert the bacteria which are sensitive to resistant bacteria? And there are two mechanisms. One of them, of course, is using pectin, which is there in apple or in the citrus peels and other things. By using that, you increase the Akkermansia, you increase the Bacteroides, and you make the sensitive intestinal bacteria into resistant bacteria. And second, using sometimes fat, saturated fat, which you don't believe that it is good for health, but that causes leakiness of the bowel to be significantly reduced. So two ways of preventing injury, and that's why sometimes people remember for taking fat with alcohol. That doesn't mean I advise taking apple and then drink, or fat and then drink, but these are just scientific basis of making bacteria change. And don't quote me outside the room. Next, please. The second approach used to be probiotic, prebiotic. I'm not going into details, but to a very recent study of synbiotics published just about 2 days ago. Next, please. And this is published in our best journal, Gastroenterology. Synbiotics alter fecal microbiome, but not liver fat or fibrosis, in a large randomized trial in NASH patients. Next, please. 1 year of giving a synbiotic, which was a fructo-oligosaccharide, bifidobacterium, and lactis, one of the lactobacilli BB12, combination of prebiotic and probiotic. What did it do? It increases the bifidobacterium, it increases the fecal bacterium, with both of which are good, and increase secondary bile acid, and one of the toxic bacteria, Oscillobacter, was markedly decreased. So at least the proof of principle is there. You can change bacteria by prolonged use of either a synbiotic and of course by diet. Next, please. The third approach is fecal microbial transplant. Next, please. This very popularly used Slovakian soldier is in a tub where he is in the fecal material, but he is still smiling and shows you with a finger that future lies with fecal microbiota transplant. Next, please. In this study, quite old and very often quoted, 26 obese people received FMT lean donor transplant, and 50% of them showed improved bacterial diversity and insulin sensitivity at 6 weeks. It lasted for 18 weeks. In the rest, there was a colonization resistance. Nobody lost weight, but it only improved bacterial diversity. So the proof of principle is FMT, which has more than one type of bacteria. Till now I showed you synbiotic or single Akkermansia studies. FMT will have, and selected group will have, very good fecal or oral ways of giving bacteria. Next, please. This is our study, proof of principle in severe alcoholic hepatitis, FMT given in 8 patients, compared to those who received no FMT antibiotics. Survival was significantly better. We have done more than 300 patients, are in the process of getting our manuscripts. FMT helps in severe liver disease, of course in obese people also. Next, please. These are potential applications of FMT in cancer therapy. The principle is you have bacteria which are likely to produce oncogenic material, metabolites, and pancreatic, colonic, and liver cancers. You change by FMT to good bacteria, maybe you can stop or even treat. Next, please. My last two slides to show you there are opportunities. Right now, we are replacing the ecosystem of the host by FMT, but this is first generation. Once we know what are the soluble factors or the bacteria, we can make a cocktail of bacteria and the cytokines and materials and give it. And finally, we can have small molecules which can be given, and the diseased bacteria or the state can be converted into a healthy state. Next, please. The market in 2022 and 2025 of the microbiome will be huge. Next, please. My last slide is the microbial therapy or intestinal microbial transplant, or giving it by any method, you can call it a shit therapy, is palatable. It is good. What it also tells us is return to your basics. We are returning in the COVID world to our basics of hand wash and hygiene. I am trying to tell you, return to basics in your food habits. What to eat and what not to eat, I have tried to tell you. When to eat, certainly be a Jenny, be a Jew, don't eat too late. How much to eat, all of us know, but we always try to ignore it. So return to basics. Gut microbiota regulates your circadian rhythm. Early to bed and early to rise makes a man healthy, wealthy, and wise. Of course, obesity you can reverse. Obesity is diet-induced obesity generally should be reversed. Diet contributes to dysbiosis, not the disease, the diet. Non-communicable diseases are probably related to diet and your gut microbiota. Fiber, pectin, inulin actually is very, very good for our life. Inulin actually neutralizes most things which comes from fiber. Protective and disease-specific bacterial therapies is the way for the next decade. Thank you for your kind attention.
Thank you so much, Dr. Sarin, for such a wonderful and enlightening talk. Indeed, this is still ignored and upcoming way of managing metabolic health and getting back to the health actually, rather than talking about the disease. So I don't think we have much time for many questions, though there are a lot of showering of questions from all over the world. So I would just take up one question, is that when we shift these patients who are very high on carbohydrate as a habit to increase in the protein, they develop diarrhea or probably change in their digestive method. So would gut microbiota be responsible for such a thing? Protein intolerance?
We are not changing them from carbohydrates. We are changing them to naturals from cooked.
Right.
So the first thing is we are changing, going back to nature, to the basics, from cooked carbohydrates to uncooked carbohydrates, and especially fibers containing carbohydrates. Yes, if you change them to pure protein, like especially supplements, they're likely to develop diarrheas because the gut is not tuned to taking direct protein. But yes, proteins in the form of legumes or the natural proteins, vegetable proteins, would always be there, helpful for us, and also proteins in the form of egg, unless there is allergy, is certainly very helpful for the body.
Thank you so much, Dr. Sarin. And I would like to thank again to the speaker, my co-moderator, Dr. Shilpa, and would like to hand over the proceedings to the organizer. Thank you so much, sir.
Thank you, Anjali.
Thank you very much. We'll end the session now.